Leaving the Earth to explore our closest neighbours is severely constrained by the cost of launching things into space. The energy required just to get into orbit is half of the energy required to get to Mars. For every 1 unit of payload that you want to launch into space, you need an additional 99 units of fuel to overcome the downwards force produced by the weight of the payload. Once escaped the confines of our planet, the difficulty of resupply means that relying on Earth to send essentials is not feasible, particularly if we want to travel to somewhere like Mars, which would include a lengthy stay on the surface in between suitable launch windows. Biology provides a unique material to overcome these challenges. It is programmable, self-replicating, and capable of self-repair. Some models have inbuilt solar functionality, whilst others use modest inorganic sources of energy. It can recycle a wide range of inputs with atomic scale precision to synthesize useful products. There is currently a collaboration between NASA and academia to engineer microbes that can harness the available wastes, volatiles, and minerals in space travel and destination planets to biomanufacture essential products. We know from the ISS astronauts that each crew member needs about 2 kilograms of food per day. For a 6-person crew, this would equate to over 10 tons of food for a 916-day Martian expedition. Aside from the launch mass, providing food with both a long shelf life and adequate nutritious value is another challenge. A current astronaut-approved superfood is spirulina, a supernutritional, protein-rich biomass made from 2 cyanobacteria. By one estimate, relying solely on spirulina could decrease launch mass by about 38%. Clearly, eating the same thing for over 3 years would not be desirable for astronauts, so current synthetic biology efforts are looking to diversify its textures and flavours. Growing crops on Mars like in The Martian would be an eventual goal if we set up a colony there, but the soil is actually full of perchlorates that can cause acute thyroid damage. Strains of the Pseudomonas clade have perchlorate detoxifying pathways, and some also have nitrogen-fixing capabilities that could enrich the soil. A Berkeley project is looking at how 2 strains fare under controlled Martian conditions, with the aim of identifying key genes and pathways involved in efficient enrichment and detoxification, to optimize or reconstruct them in other organisms. Headaches are a really common complaint in space. Pharmaceuticals, however, can be problematic to supply because radiation can accelerate their expiry. It is possible to produce paracetamol in E. coli using a modified chorismate pathway with a gene from fungi to catalyze one of the substrates. E. coli, however, is not a space-appropriate organism. But reconstructing this pathway in an autotroph like cyanobacteria or even spirulina could be a viable solution. Just a few days of paracetamol production could completely replenish irradiated stocks of this pharmaceutical. While organisms like E. coli and Bacillus subtilis have the most synthetic biology tools available for engineering, they are less suited to resource utilization off-planet than autotrophs. A way to get around this is by employing feedstock-producing intermediate organisms to harness the available resources into something more usable for production microbes. One iGEM project that contributed to this idea was the collaboration between Stanford and Brown, known as PowerCell. This is a diazotrophic cyanobacteria, engineered with the functionality to make and secrete sucrose. With energy from the sun, PowerCell could conceivably convert Martian resources into usable feedstock for production organisms. This project was actually selected to be launched into low Earth orbit as part of the Eucropolis satellite, designed to stimulate Martian and lunar gravity each for 6 months. As part of this payload, PowerCell was paired with an endospore-forming potential production organism, Bacillus subtilis, engineered to express a particular pigment when metabolizing sucrose. Along with the recent work done on the ISS, this was one of the first experiments to test the effects of gravity on protein expression and cellular functioning in microecosystems, and the data will have important implications for designing biological systems for space travel. Humans have always depended on a plethora of microorganisms. Once we leave the petri dish of Earth, there is no reason why we shouldn't continue to depend on them, especially when we have the tools to mold them with synthetic biology. Ultimately, the technologies that help us to live in space will undoubtedly revolutionize life for the humans who remain on Earth.